We are recording. It's 8 o'clock. I'll just call out your name if you're not here. So if you hear your name, let me know, because you shouldn't. Carr. Smith. Davis. Zoe Davis. Stanley. Whitaker. Sims. Anybody else? Anybody here wearing WVSU gear? Brock. Anybody else? All right. So if Brock wants to take the picture, and somebody wants to or be in the picture, and if somebody wants to take it, takes it. Obviously, you get an extra credit too. But we'll cross that bridge at the end of the class. For announcements, being a dead horse, but man, it's halfway through the semester. Some of you guys really need help. Like I'm grading the labs and stuff, and just really need help. But that's what I'm here for. Remember, you don't even have to come to my office for office hours. You can just show up online, and I can help you. So please let me help you. Unless you don't care, in which case, whatever, I get it. You know, there's other things in life other than biology. But if you're worried about your grade and care about it, please let me help you. Anyway, so that's that. I'll remind you too, the exam will be on Monday, just like it was last time. If you miss it, you just miss it, that's it. If you want to take it early, let me know. Another thing I haven't told you yet, though, is that I will be traveling this weekend. So if you feel like you might need help between now and then, try to reach me before Friday, because after class on Friday, or after office hours on Friday, I'm going to hit the road. So if you need me, let me know early. I think that's it. Other stuff I've just said way too much. I'm not going to repeat myself. Are there any questions about anything in general? Independent work, lab, attendance, exams? I'm going to look up the date to drop the class and share that with you guys, because some of you guys should really highly consider it. You should know. But again, if you don't know, you can maybe during office hours and we can talk about your progress, and I can talk about how come up with a plan to help you, and you get those extra 2 points just for showing up to my office hours, online or in person, so I can help you. Anyway, if there's no questions, let's jump back into chapter 8. To remind you, this chapter will not be on this coming up exam, the one that's going to be on Monday, because that exam is going to go to chapters 5, 6, and 7, which is all about energy, right? This next exam is going to be about chapters 8, 10, and 13, which in a sense is basically all about reproduction. We'll get into DNA and stuff like that too, but so far in this chapter, we've talked about what cellular reproduction accomplishes. And basically what we learned there was that there's asexual reproduction versus sexual reproduction. And asexual reproduction is done via mitosis. Sexual reproduction is done via meiosis. That's the big thing you learned in that first main bullet point. Then we started getting into the cell cycles. And we talked about eukaryotic chromosomes. And back on that, basically you need to know what a chromosome is. I told you some other stuff in that bullet point that'll be important later in the semester, so I'm not going to talk about it right now. Then we talked about duplicated chromosomes, how like you have just that one chromosome, but then during the S phase of interphase, it has to duplicate itself, right? Because it's getting ready for the whole cell to reproduce, including the stuff in the nucleus. And when that happens, you have two chromosomes attached to each other, two perfect copies of themselves. And at that point, those are called sister chromatids. And they're attached at the centromere. And then, during mitosis, they actually split. Then we talked about the cell cycle. So the whole cell cycle is basically the life of a cell, right? From the moment that it was created from another splitting cell that came before it, until the moment it itself splits, that is the entire cell cycle. And you needed to know that the entire cell cycle is basically broken down into two main stages, interphase and the mitotic phase, right? And you also needed to know that the cell cycle is mostly spent in interphase. The book was very specific. It said 90% of the time your cells are in interphase. But you just need to know the majority of it is in interphase. And then when it comes to interphase, we broke that down into 3 phases, right? Interphase itself is broken down into G1, and then S phase, and then G2. And I told you do need to know those in order, and that you do need to know that S1 is the most important as far as this conversation is concerned, because that's when the duplication of the chromosomes happen, and you need to know that. Then we get into the other part, right? So we're looking at the entire cell cycle. Again, we just got done talking about interphase. Then you look at the mitotic phase. The mitotic phase itself is broken down into two parts. There's mitosis and then cytokinesis. And those things overlap, so it's not like G1 and then it's not like G1 and then S phase and then G2, right? Those are distinct. These things overlap. So mitosis, as it's ending, cytokinesis is starting, right? So those are the two stages of the mitotic stage, right? And then mitosis itself is broken down into 4 stages, and that's where we left off. That's what we're about to talk about, the 4 stages of mitosis. The first word for attendance, sorry I didn't say that earlier. Let's say the first word for attendance is out, because it's on the screen, so it's a nice easy one to start out. The first word was out. Make sure you send me your words before 9 a.m. Hopefully this time I'll actually be able to do questions. Well, not hopefully for you guys. I guess it's easier to just send me your words. But anyway, first word for attendance is out. Let's jump into this, mitosis and cytokinesis.
I have a quick question.
Yes.
When it goes through the interphase and then after it splits, does that one cell that split does something else and then the other one, does it go back through the...
Good question. So if you look at I'll just show you the picture. So what's happening here is when the one cell splits, it's now 2 new cells, essentially.
Okay.
And we don't get into exactly how, I mean, we'll touch on that a little bit when we talk about what happens with the chromosomes. But for the rest of it, we don't even talk about it. Just know that, in a sense, this right here is the death of the singular cell in a way, because it's now split into 2 new cells. Of course, those 2 new cells are literally half of the old cell. This one's half the old cell, and this one's half the old cell. So it still carries on, so to speak. But that is the end of the life cycle of that cell because it no longer technically exists. It would be almost as if humans would be produced that way. It's like, all right, well, it's time for you to go. And we cut you in half. And then, like, one half of you plopped off, another half of the body, the other half plopped off, and your half of the body, and your 2 little clones went their separate ways. Does that make sense? Okay. It's a good question. Not that it would be on the exam, but yes, good question. So, you don't need to write this down yet if you don't want to, because we're going to talk about each of these in detail. But the 4 distinct phases of mitosis are prophase, metaphase, anaphase, telophase. You will need to know that. There will be a question on the exam that asks you what the 4 stages are and what order do they go in, and that is the order that they occur. There'll be another question that looks like that isn't, though. There'll be another question that says, in this stage, the chromosomes do this. In this stage, the chromosomes do that. In this stage, the chromosomes do this, and it'll basically fill in the blank. So make sure you don't mess those 2 up. There's 2 questions where the choices are going to be prophase, metaphase, anaphase, telophase. One of them is going to say, put them in order. The other one's going to say, in which stage does this happen? In which stage does that happen? So as we learn this, keep that in mind. That's what a lot of what the test is going to be about. You need to know what happens in each stage. I should also point out that even though your book says it's distinct, just for your own knowledge, not for testing purposes, but for your own knowledge, it's not like this all happens like, it's prophase and then all of a sudden, bam, it's metaphase. And then all of a sudden, bam, it's anaphase. It doesn't really happen that way. Prophase kind of eases into metaphase. Metaphase kind of eases into anaphase. And for some of you who've already done the lab and got some questions wrong, and I'm using quotation marks for a reason, you're aware of this because the biggest mistake I saw was people were picking cells that were in anaphase and saying that it was in metaphase. Let me rephrase that. People were picking cells that I thought looked like they were in anaphase, and they thought they looked like they were metaphase. But because they kind of go smoothly into each other, it's debatable. So if you got that question wrong, you'll know I left you that comment and said, all right, to me that looks like anaphase. If you can explain why you think that's metaphase, then you can get the point. So again, for your own knowledge, these things do kind of ease into each other. As a matter of fact, if you were to look this up, and you can print a pen org if you want, this, you guys are getting the Cliff Notes version. You guys are getting the Bio 101 version. There's actually stages in between this, like pro-metaphase or pro-anaphase, right? There are actually, it's broken down even more. We just don't get into it. But anyway, any questions about this slide? So far, the only thing I told you as far as the exam is concerned is you need to know these 4 stages, and you need to know that they go in that order. But, actually, before I jump into this. Let me also point out, I said this on Monday, a lot of stuff obviously has to happen when a cell duplicates. First of all, it has to double everything it has, including the chromosomes. Then it literally has to cut itself in half when you think about it. So it's very complicated. But all we're focused on for the most part is what happens to the chromosomes. So mitosis, especially in this course, is the story of what happens to the chromosomes. It's all about the chromosomes. So again, if you're interested in it, you can look it up for independent work. What else? Like when we talk about prophase, and I'm telling you this thing's happening to the nucleus and this thing's happening to the chromosomes, you could look it up if you want to say, well, what else is happening in prophase that we didn't talk about? Anyway, like I said, this is all about the chromosomes and how they move around. So before we can talk about that, I want to introduce you to something called a mitotic spindle. That is the thing that attaches to the chromosomes and moves them around. So this whole thing is about how the chromosomes are moving around. Obviously, we need to know how they move around, and that is the answer to the mitotic spindle. I'm going to put a question mark. I probably won't ask you to define a mitotic spindle, but I'm definitely going to use that word a lot. So you definitely need to know what it is when I use it. But yeah, mitotic spindle is that thing that moves the chromosomes around. Yeah, it's football shaped. I don't care about that. I'm not going to ask you. Definitely won't ask you what shape it is. Yes, it's made of microtubules. But we skipped chapter 4, and I don't even remember if chapter 4 tells you what microtubules are. So you don't even know, you need to know that by name. I'll just tell you microtubules are literally the things that come out and attach to the chromosomes. You can almost think of them as ropes, if you want. And again, like I said, those are the things that are guiding the chromosomes. Those are the things that are moving the chromosomes around, because this is all about what happens to the chromosomes. And when you define prophase, metaphase, anaphase, and telophase, that's how the question's going to go. In which stage are the chromosomes being pulled towards the middle? And what stage are the chromosomes being pulled apart? Things like that. So what is going on with the mitotic spindle will be important to define each of the 4 stages. So any questions about what a mitotic spindle is? Okay. Look at this. Let's talk about this for a second. Now, let's say, this is this, right? Let's say there's, what is this, 1, 2, 3, 9 balls of yarn. Imagine this is also 9 balls of yarn, right? But this is the same 9 balls of yarn that have been unrolled, right? They're just a tangled mess. But imagine if I said, all right, and then let's imagine like for every foot, there's a number on one of these. It's hard to see, but it's number 1, and then a foot later, it's number 2, and then a foot later, number 3. All the way, starting from number 1, all the way down to number 1 million or whatever. Now, let's say I said, I need you to find number 26. And let's say they weren't in order. How about that? What do you think would be easier to find something on the string? Here or here?
Put together.
This one?
To find the number?
Yeah, let's say somewhere on some string is a number that I need you to find.
I would say when it's spread out.
Spread out, right? This is nice and neat, so I can see why your brain might go there. But think about it, if it's like in the middle of that one, you've got to unravel it, right? So to get to the number, you have to unravel it. Well, you can think of those as chromosomes, and you can think of the number as genes. So if we need a gene, and your book or your cell needs to read that gene and produce whatever protein that gene says to produce, then your chromosomes actually need to look like this. That way they can get to it. Because if your body needs a gene on that chromosome, well, the first thing it's going to do is unravel it to be able to get to it. Does that make sense? When it's all raveled up, you can't get to it. That's why in all of Interphase, your chromosomes look like this. And we've talked about that, how they look all squiggly, and they're not even visible to the naked eye because they're all a squiggly, uncondensed mess. Well, that's why. Because your book, I don't know why I keep saying your book, your cells need to get to your chromosomes to be able to read the genes, right? In all of Interphase. Now, when you go through mitosis, that's a different story. Mitosis, we're trying to split things up. So now let's say we had a race between you two, right? And I said, all right, I'm going to put this yarn right here in the middle, and I want you to put half of them here, half of them here. And she gets this one. She gets the balls. And then he gets the big squiggly mess. I said, all right, I'm going to put all this yarn right here. And I want you to separate it. Half here on that table and half there on that table. Who do you think would win the race? The person with the balls or the person with the big squiggly mess? If the race was, I need you to separate them completely in half, half on that side, half on that side.
Do I have to unroll them?
No.
Oh,
Balls, yeah, the balls.
Yeah, the balls, right? Because they're just bloop, you just move them across. Of course, it's kind of hard to do, cut 9 in half. I need to find a picture that has like 10, but yeah. That's why before mitosis, your chromosomes coil up. Because one, you don't really need to read the genes anymore like you used to in that space, but you also need to be able to easily move them. So that's just another way of remembering that during mitosis, your chromosomes are condensed up. And that's why, because it's easier to move them. And of course, they don't always need to be rolled up because at other times you need to be able to get to the genes. Ooh, so that'd be the second word for attendance. The stuff you see right down there, yarn. So any questions about that concept before we jump into it? All right, well, that being said, let's jump into the first stage of mitosis. Also, I want to point out, a lot of times when you read stuff, and this is just a reminder, you're going to read how it goes from interphase to prophase, right? And it does, right? Because interphase is the other part of the cell cycle. But keep in mind, interphase is not a part of mitosis. So when you get a question that says, what are the stages of mitosis in order, and then interphase is in there, that's not the right answer. If it has interphase, don't choose that one. Because yes, interphase comes before prophase, but it's not a stage of mitosis. Like if I were to ask you, what's the first year of your 20s? And then you said, well, it goes 19, and then 20, and 21. Well, you'd be incorrect. Yes, it does go 19, 20, and 21, right? But 19 is not part of your 20s. Same thing here. A lot of times when you talk about mitosis, you do talk about interphase and then prophase, metaphase, interphase, telophase. But interphase is not part of mitosis, so please don't mess that up. Anyway, how do you define prophase? This is how the question is going to go. On the exam, you're probably going to get all these things, or some of these things. I'm going to say, in which stage do, list all these things. And then you should recognize this as prophase. So if you're not a good test taker, hopefully that eases your mind a little bit. The way that this is not going to go like this, where I say, list everything that happens in prophase, and you have to write everything out. Right? You don't have to do that. You're going to get all of these, or some of these, and you just need to recognize it as prophase. That being said, let's talk about the first thing, which we already did. In prophase, the chromosomes coil up. They're always coiled through all of mitosis, but this is the phase where they finally coil up, right? Because we're going from interphase, where they're not coiled up, to prophase. So they coil up, and we already talked about why, right? Because it's easier to move them around. At this point, they still exist as sister chromatids that are joined at the centromere, which is basically saying, remember all that stuff we said that happened in the S phase of interphase? Yeah, it's still that way, right? They're still together. Now going back to the example I used for the balls of yarn. I'm going to back up for a second, and I'll come back to this. But yes, we've got these balls of yarn. Sorry to pick on you, but you're up front. Now imagine I had those in a Kroger bag, and I had it tied up, and I just asked you to do the same thing. I said, all right, I need you to take these balls and move them half on this side, half on that side. Go. What would you do? What would be the first thing you do if I handed you those in a tied up Kroger bag?
be competitive, I would rip open the Kroger bag.
There you go. You rip it open. Either way, you get them out of the bag, right? Well, the same thing happens here. Right? If you're going to move chromosomes to other sides of the cell, which is what you're going to do, right? Because you need to split the cell in half and have two cells. You're going to need to basically get rid of the bag that's holding it. So the nuclear envelope breaks into pieces. Or even, I'm not going to write this, it's really hard to write on the screen, but I'll even abbreviate that and say it disappears or it goes away. So when you memorize, don't memorize that verbatim. Just understand that in prophase, the thing that holds them together, which is the nucleus, right? That's where they all stay. It goes away, right? It disappears, disintegrates, whatever you want to call it. Because, again, we need to move them to different sides of the cell. So if they're all stuck inside the nucleus, that's not going to work. I'll also put a star next to this because, no, I don't want to say that. Never mind. So again, chromosomes have coiled up so we can move them around. They're obviously still joined together. The chromatids, the sister chromatids are still joined together. The nuclear envelope's broken into pieces so we can move around the chromosomes. But how do we move around the chromosomes? Well, we need to attach it to the thing that moves them around. So that's the other thing that happens. The spindle attaches to the centromeres. That's pretty specific. I might be less specific on the exam and say the spindle is attached to the CH, I'm gonna abbreviate again, the chromosomes, because that says the same thing, right? The spindles are attaching to the chromosomes, and to remind you, the centromeres are just the middle part. So horrible. There's my sister chromatids, there's the centromere, right? So those are the chromosomes, but technically that's where the spindle attaches. So again, that question could be asked two different ways. Or said to, that bullet point could be said two different ways. The spindle attaches to the centromere, which is very specific, or a little bit less specific, the spindle attaches to the sister chromatids. Which all makes sense, right? This is all logical. If we're going to move chromosomes from one side of the cell to the other, they do need to coil up. They have to be together. But first, you do need to get rid of the envelope. The thing that moves them does need to attach, right? And then the very last part of it, and this should make sense too, because the point is to, well, this actually is not that intuitive. There will be when you get the whole picture. Before they can split to different sides of the cell, because right now they're all over the place, first they have to meet in the middle, and then they split to the different sides of the cell. So that explains the last bullet point. After the spindle attaches, then the chromosomes start moving towards the center of the cell. Now, one of the questions might be really, really simple or simplified, where instead of asking all the different things that happen in each stage, it might just simply be asking where are the chromosomes going. So if that were the case, just remember in prophase, the chromosomes are moving towards the middle of the cell. So actually I would memorize that first and then worry about all these details. So in prophase, the chromosomes start moving towards the middle of the cell. Because again, when we talk about mitosis, even though so many other things are happening, this is really a story of what the chromosomes are doing. Let me ask you this. This should be logical if you think about it. Don't overthink about it, but just think. If I'm telling you that what defines prophase is that the chromosomes are moving towards the center of the cell, then what do you think must define the next stage? If they're moving towards the center of the cell in prophase, then what do you think defines the next phase? Don't overthink it. What do you think the chromosomes do in the next stage? Yeah, if prophase, they're moving towards the center of the cell, then logically the next step is they made it, right? It's this that simple. In this phase, they're moving towards the center of the cell. In the next phase, they made it. We'll talk about that here in a second. Here's some pictures of prophase, also interphase. There's the big squiggly mess, right? There's the yarn that's unballed so you can get to all the genes because you're in interphase, right? So you need to use all those genes. Then you finally get into prophase, right? So we're getting into mitosis. The chromosomes coil up. The centromeres attach to them so they can move around, or that's the mitotic spindle. The centromeres are moving on the way to attach. Then they get attached. Then they start moving towards the center of the cell. You can see the nuclear envelope has disappeared. All those things we talked about. And at this point, even though you can't really see it, these things are all over the place, but right now they're moving towards the center of the cell, right? And as they're moving towards the center of the cell, that whole time they're pretty much in prophase until they get to the center of the cell, which at that point they are in metaphase. So yes, I'm about to tell you a lot about metaphase, but really metaphase is when the chromosomes get to the middle of the cell or the center of the cell, however you want to pronounce it or describe it. Again, when you're studying this, don't do it verbatim because there's so many different ways to describe the same thing. So that is metaphase. Excuse me. And the way I always remembered this when I was a student is because the name almost describes it. Of course, meta means something different. That's a Greek word, but you could almost think that it's like, oh yeah, that's when the chromosomes met in the middle. Right? So if you remember that, metaphase is when they met in the middle. Again, that's the biggest thing. For studying, remember that, and then we'll remember all the details I'm about to tell you. So in prophase, they were going towards the middle. Metaphase, boom, they made it to the middle. At this point, obviously, I don't know why your book even puts this on there, so I'm going to exit out. But the spindle is obviously fully formed because if we've moved all those chromosomes to the middle and the thing that moves them is the spindle, of course the spindle is fully formed, but your book included it, so did I. Like I said, the big point here, the most important thing as far as we're concerned, is the centromeres line up between the two poles of the spindle. Like I said, you don't have to memorize it verbatim, because another way of saying this is the easier way that I said it. The sister chromatids met in the middle, or the chromosomes, even though that's not technically the proper term. You could, sister chromatids is better, but the chromosomes are now in the middle of the cell, right? But yeah, that's the same way of saying everything we've been saying. And I might say it any number of ways. What I won't do is give you a bunch of things that look like this, and you have to pick out the more correct one. It won't be anything like that. Basically, if it says anything about lining up in the middle, that's metaphase. Your other options are going to be it's moving towards the middle, or it's moving away from the middle. And that will not be the right answer. If it's in the middle, that is metaphase. Here's where it gets a little bit different. So they meet in the middle, and then once they've all lined up in the middle, I'm going to put a little dash to here. Not that you need to separate these two in your mind, but in a way they're different things, right? So they meet in the middle, and then once they're all met in the middle, then those spindles start pulling them in the opposite direction. It's like a tug of war. Now, because it's a tug of war and they're evenly matched, all the chromosomes stay in the middle. Now I'm going to put that, I'm going to put an X through that because you don't necessarily need to know that for the exam. I'm just trying to explain things. Imagine if you've ever, I'm sure you guys have seen people play tug of war. Imagine if you had two perfectly matched teams. That's what the way it would be, right? The middle of the rope would be in the middle, right? Because they're perfectly matched. However, this is not really like tug of war because these ropes, that's not one continuous rope, right? That's technically a rope attached to half of a chromosome, sister chromatid. That rope's attached to the other half. So those things are pulling on something that could eventually rip, is what I'm saying. So again, let's look at this logically. In metaphase, they met in the middle. Now these things are trying to pull them apart. So if metaphase ends, right, the ending of metaphase is these things getting pulled, what do you think actually begins the next phase? What's the next logical step? If these things are getting pulled, what do you think defines, what do you think the chromosomes do on the next step? And this is all very logical, so don't overthink it.
Break apart.
Perfect. They're breaking apart, right? So right now they're getting pulled, but they're holding on. But as soon as they don't hold on anymore, as soon as they're broken apart, that is the next phase. That is anaphase. But let me give you some pictures of metaphase first. Oh, there it is, metaphase. You can see how they're all. You know what, too? I can see. No, I'll get into that later. I was thinking about lab when I looked at this, but I'll talk to the people who got that question wrong. But anyway, that's what metaphase looks like. Let's talk about the next one, which is anaphase. At that point, the sister chromatids are now separate, right? And again, that's logical because the way we ended the last stage is they were all getting pulled. So once they separate, that is then called anaphase. And that's, I'm going to circle it because that's the most important thing you need to know for the exam, anyway. Because again, the most important thing for the exam is what's happening to the chromosomes. So an anaphase is defined by the fact that they have all separated. Now for your own knowledge, as you might imagine, that usually doesn't all happen at once. It's not like all the sister chromatids, boom, separated once. Probably one separates, and then another one, and then another one, and another one. And for your own knowledge, it's not technically anaphase until they've all done that. But you don't need to know that. For your mind, you can just think, all right, once they're separated, once they're first separated, that's what we call anaphase. And at this point, technically, we're not talking about sister chromatids anymore, right? Because when they're attached, when those two perfect copies are attached to each other, they're called sister chromatids. And like I said, way back when we were talking about S phase, or when we were talking about chromosome duplication, I said once they get pulled apart, once they're separated, then they're considered full-fledged chromosomes. I will put an X to that, not because you don't need to know it necessarily, but because I'm not going to ask you that on the exam. Just know that an anaphase, again, that's the point where the sister chromatids have separated and they're now just regular old full-blown chromosomes. And I'll put another dash. So we talked about what defined anaphase, which is the chromosomes finally separated. Here we go again. You should see a pattern here. Now I'm going to tell you what they're starting to do. They're starting to move towards opposite ends of the opposite ends of the cell, opposite poles of the cell, opposite sides of the cell, however you want to say it. they're moving apart, which is logical, right? So then think about this logically. If they're moving towards the opposite poles of the cell, an anaphase, how do we define the next phase? What is the thing the chromosomes are doing in the next stage that makes it the next stage? If in this one, they're moving towards opposite poles of the cell. I'll give you a refresher. It's a good time to just repeat what I said because it'll help you remember it. In prophase, they're moving towards the middle of the cell. And then in anaphase, they got there. And then in anaphase, they were getting, excuse me. In prophase, they were moving towards the middle of the cell, right? And then metaphase, boom, they got there. That's how we define it. And then in metaphase, right, they were starting to get pulled. They were getting pulled. And then finally, boom, they pulled apart. And that was anaphase. And now in anaphase, they're moving towards the opposite ends of the cell, opposite poles of the cell, however you want to say it. So what do you think defines, what is it that makes the next stage? If in this one, they're moving towards the opposite ends of the cell, then how would we define the next stage? They made it. It's basically that simple, right? Basically, and that's what you said, right? If this in anaphase, they're moving towards opposite ends of the cell, then guess what? In the next stage, they make it. Of course, more happens than that, but yeah, they make it. And we'll talk about that when we get there, but also I want to point out that the poles are also pushed farther apart, which elongates the cell. I'm not going to ask you that, but think about it. Cells are generally round, right? So we're dealing with something that looks better than that. But that's what they normally look like, but we're going to cut this thing in half eventually, right? So what happens is, in anaphase, what we want is a big oval, right? That's horrible. So we can eventually cut it in half. So if we're going from a circle to an oval, at some point in this process, obviously it needs to start doing that. And for your own knowledge, I'm not going to get this deep on the exam, but in anaphase, that is when that starts happening. So this whole time, we've had a nice little spherical cell, and now in anaphase, it finally starts to get oblong and move towards the opposite ends. But again, the most important thing here is what's happening to the chromosomes. All right, any questions about anaphase? Okay, here's a picture of anaphase. You can see in this picture, it's hard to see for you guys. Online, you might be able to see it better, but I should have mentioned this earlier. Not that it matters too much, you won't be tested on it, but the green part, that's the spindles. The blue part, that's the chromosomes. Obviously, that's colored to make it look a little bit visible. Anyway, in anaphase, the chromosomes were moving towards opposite ends of the pole. So then the next and final phase is telophase, and that's when they finally made it to the opposite ends of the pole. So again, I'll circle that just to remind you that that's the most important thing in our discussion is what happens to the chromosomes. So in telophase, they have reached opposite ends of the pole. Opposite ends of the cell. Opposite sides of the cell. However you want to say it. So now that that's happened, as far as the chromosomes are concerned, the job is done, right? They were duplicated in S phase, but we separated those so we have full-fledged chromosomes. We separated them all the way to the opposite ends of the cell. Now we just need to reverse what we already did. So we need to put the nuclear envelope back, right? Because we no longer need to move them around. So, all right, let's get the nuclear envelope back. The chromosomes can now uncoil, right? Because again, we need to use them to read genes. They don't need to move around anymore, so they don't need to be coiled up. Like I said, we don't need to move them around anymore, right? So the thing that moves them around, we don't need that anymore, so the spindle disappears. So in shorthand, for studying prophase or telophase, you can just remember chromosomes are at opposite ends of the cell and reverse the prophase. And hopefully you've already memorized when you're studying. Hopefully you've already memorized what happens in prophase. At this point, again, telophase is the last part, right? So at this point, telophase is over, therefore mitosis is over. And while telophase is going on, right, while the nuclear envelope is forming and the chromosomes are uncoiling and the spindle is disappearing, while that's going on, cytokinesis usually starts. Because like I said when we started this conversation, the mitotic phase is broken down into mitosis. Do it like this, since it's the way it looks like on your book. That's interphase. This is the mitotic phase. I told you the mitotic phase was broken down into mitosis and cytokinesis. And I also said that they kind of interlap, right? So there's a point where it's going through both, and that's right here, right? Before the exam, you don't necessarily, I'm just telling you that for your own knowledge. For the exam, you don't necessarily need to memorize the fact that they overlap. In your brain, for the exam, you can just think telophase, and then cytokinesis, how to combine two real words to make one other real word that has nothing to do with biology. Anyway, yeah, telophase and then cytokinesis. And we're about to talk about what cytokinesis is, but for this slide, you just need to know what telophase is. So any questions about what telophase is? And again, like I said, before we start talking about all these things, a lot of stuff is happening in your cells. The only thing we're talking about is what's happening to your chromosomes. So if you're curious, you can look that up for independent work. What else is going on in your cells when you're going through mitosis? Anyway, the next word for attendance will be, I'm going to circle it again. So again, if you're listening to this and not watching, open your eyes, even if it's just for a second, and look at that word. Consistently, there's people who always miss one word every day, and it's always the word that I circle instead of say. So again, if you're online right now, open your eyes and look. It's the next attendance word. Anyway, any questions about telophase?
This is the second word, right?
Nope, it's not the second word. There's more than that. Good question. But I think it's the third word. Yeah, I think it's the third word. I know it's not the second. Any other questions? Any questions about telophase? Okay. Let's talk about cytokinesis, the thing that's ending all of this. Again, like your book says, it usually begins with telophase. To be honest with you, I've read sources that say otherwise. If you want to look that up for independent work, you can. Like what are some other sources? What do other sources say about when cytokinesis begins? Of course, for you guys, you're responsible for what your book says. So if I were to ask it, the answer for you guys would be begins with telophase. But again, I know we'll go ahead and talk about it. The reason I think you get different answers depending on where you look is because remember when I was talking about anaphase, I said the cell itself starts going from circular to oblong, right, to more oval. And it's doing that in preparation for what cytokinesis is, which is cutting the cell in half. So by that definition, you could say that it's starting in anaphase, because that is when it's starting to become more oblong. So some sources do say that. And again, you can still look it up even though I gave it to you. You just include your source. Anyway, but the way your book defines it as, it starts in telophase. Again, I'll put a next to that to remind you. You don't necessarily need to know that for the exam. That's just for your knowledge. What does cytokinesis do? Well, this is the important part. This is what you need to know. If anything, that's the most important part of this slide. It divides the cytoplasm into two cells. It cuts the cell in half. It makes you go from one cell to two cells. However you want to say it, this is the thing that finally does it. This is the thing that ends it, right? This whole time we keep talking about we're trying to go from one cell to two cells. Well, this is the thing that finally does it. Right? You went through mitosis, your nuclear envelope and everything in it doubled, right? So your chromosomes doubled and went their separate ways. So at this point, before cytokinesis happens, you have one cell with two nuclei, right? And then finally, it gets cut in half, and then you have two cells, each with their own nuclei. This process is slightly different between plants and animal cells. Let's talk about that. As you already know, and we haven't talked about too much of the difference between plant and animal cells, but you do know plant cells have cell walls, so keep that in the back of your mind. Let's talk about animal cells first. In animal cells, cytokinesis is known as cleavage, and it begins with the appearance of a cleavage furrow, which is an indentation at the equator of the cell. We'll put an X to this whole thing. You don't necessarily need to know that. You definitely don't need to know what's called cleavage. You don't need to know all these fancy words. Basically, it starts caving in on itself, so to speak. It starts cutting itself in half right towards the middle. To me, that looks like if Pac-Man were real and there was a picture of it. It's like an old Pac-Man. But anyway, that's what that is. Here's another picture of it. there's the cleavage furrow. There's a drawing of it. Again, cytokinesis is just cutting the cell in half. And basically, in animal cells, it's just that simple. You're cutting the cell in half. So really, if you get any questions about the difference between animal cells and plant cells, I won't even ask you about the animal cells because it's just getting cut in half. You'll just need to know the special thing that happens in plant cells, which is what we're about to talk about. Are there any questions about how it happens in animal cells? Okay. And of course, as you can imagine, even though I just said this in like 30 seconds, this is actually a pretty complicated process. Your book doesn't get that complicated. If you want to look into it for independent work and tell me some of the stuff that's happening, exactly how does that cleavage furrow form? You know, what's happening to all the stuff in the cells in that area? You can look it up if you want to. All right, if there's no questions about the animal cells, let's talk about the one that's different, which is the plant cells. As you already know, one of the ways that plant cells are different from animal cells is they have cell walls. Therefore, it's not as simple as just cutting the cell in half with the membrane. You also have to deal with a cell wall. And that's basically what happens. So that's the only difference, right? So what's the difference between plant cells and animal cells for this? Well, you have to build a new wall. That's it. Technically, it's called a cell plate, but I'm not going to ask you what a cell plate is. Technically, it's vesicles that do it. So little bubbles inside the cell that carries the material to where it needs to go. That's how it happens, but you don't need to know that. You can see here in this picture, it's not like it starts on one end and works its way to the other, right? It just starts all over, and they just all kind of connect. Which makes sense, right? If you were to build a wall, if you were going to build a wall on the border of the United States and Canada, to keep the Canadians out, or maybe keep us out of Canada, you wouldn't just go from like Washington to Maine, right? You would start all over, right? You have some people working in Washington, some people working in Montana, some people working in North Dakota, some people working in New York, some people working in Maine, right? You're all working on the wall together at the same time, and eventually they'll all link up. It wouldn't make sense to just go from one end to the next. Anyway, any questions about cytokinesis in plant cells? All right. Let's see, 8 minutes. That's enough time to talk about this. Speaking of mitosis and all these things that happens, sometimes things go wrong. And that's what cancer is, basically. So I'm about to give you a basic introduction to what cancer is. Obviously, cancer is very different. It's a very complicated diseases, but you're going to learn the basics right now. For plants and animals to grow and maintain tissue, so that includes you, right? You're an animal. For you to just live and replace cells that die or to grow, which most of us aren't growing anymore, you have to control the timing of cell division. Which is basically, I'll remind you what we said at the beginning of this chapter, is that you, as sexually reproducing organisms, do mitosis and meiosis, right? So when you're trying to make another human being, that's the only time meiosis is important. But right now, as you sit here, you are going through mitosis. You are reproducing, right? Because when cells die, you need to replace them. When some are dying, sometimes you need to replace them. Or when you grow, you need to make new cells. So again, you are doing this right now. But obviously, you can't just have that constantly happening. Otherwise, it wouldn't be good for a number of reasons that we don't have time to talk about. So what has to happen is this. Again, you have to control the timing of cell division. So the cell cycle isn't just constantly going. There's start and stop signals throughout this whole thing. And what controls that? Specialized proteins. So it's all these different proteins involved that say stop, go, basically. Because again, I do the cell cycle here. Yes, it goes from the beginning of interphase and then through the mitotic phase, and they split and it goes again. But it's not like a clock. It's not constantly going. It might go to G1 and stop, and then something tells it, all right, go to S phase and then stop. And then something says, all right, now go to G2. All right, now stop, right? There's things involved that say go and stop, because it's not like a clock constantly going. And a lot of it has to do with what's going on in your body, right? Because if your cell has a purpose, like a kidney cell, for example, it's being controlled by other things. Like we talked about in chapter 1, your body sends signals, right? There's information flow. So How does it do that? Well, again, there's specialized proteins involved. So any questions so far? To summarize everything that's on this slide, what you might need to know for the exam, if anything, is, again, we need to control the timing of cell division. And we have basically on-off switches. Your cell has on-off switches. Even if you don't remember any of the other details, then that should suffice. Because again, I'm just trying to explain to you what cancer basically is, right? Before I can do that, you need to know this simple fact. The cell cycle has to be controlled because it's not constantly going. So we have built-in on-off switches. So cancer is a disease of the cell cycle. So cancer is basically where our on-off switches are broken. Another way of saying that is cancer cells don't respond normally to the control system, i.e. the on-off switches. And because of that, they divide excessively. So in cancer's case, the cell cycle does continue to go and go and go without the on-off switches. That's basically what cancer is. That's the basic answer to what cancer is. And when that happens, these cancer cells then may invade other tissues. So very often when you have a cancer and it goes untreated, it's not going to stay where it started. It'll start going into other tissues. So how does all this begin? That's this bullet point right here. The abnormal behavior begins when cells undergo genetic changes in genes that encode for proteins in the cell cycle control system. I'm only putting an X to this because I'm not going to tell you on the exam. That is something you should know for your everyday life, right? This is one of the more important things that you're learning in Bio 101 because cancer affects all of us. Either you're at risk for some kind of cancer or you know somebody who has cancer. One day in your life, unfortunately, you're probably going to have to deal with cancer. Hopefully not you, hopefully not somebody you love, but at least somebody you know. I've had to deal with a lot of people I know who've had cancer. But anyway, now you know how, right? So the on-off switch is broken. And the more scientific way of saying that is, like I told you, the proteins involved, right? So there's something wrong with the proteins involved with the on and off switch. And as you're going to learn in the next two chapters, well, in the upcoming chapters, that's how genes work, right? Genes are the code to say, build this protein, build this protein. So if you mess up that gene that says build this protein, and that protein is part of the on-off switch, then your on-off switch is going to get messed up. So for example, that's how smoking might give you cancer. So you have all these bad chemicals when you smoke. Some of them mess up your genes, right? Your DNA, they mess it up, they change the code, and then your body's now making this mutated version of this protein, and because it's mutated, it doesn't work properly, and because your on-off switch isn't working properly, boom, you have cancer. Yeah, so that's the basics. Any questions so far? Here's another very important slide for your everyday life. I'm going to put a big X to it because I'm not going to ask you these questions on the exam. But cancer cells, you know, when they grow out of control, they form what's called tumors. I'm sure you all know what tumors are. Those are abnormally growing masses of cells. Now, not all tumors are cancer. You can look that up for independent work if you want. But cancer will eventually form tumors. If it remains in the original site, we call that a benign tumor. If it goes elsewhere, we call it malignant. So if you ever hear somebody say, oh, it's malignant, that's because it's spreading. It spreads into neighboring tissues and other parts of the body, which then makes new tumors. And the way it's deadly is that interferes with the organ function, right? So, you know, if it gets into your colon, it keeps your colon from doing what colons are supposed to do, then it can kill you. So individuals that have these malignant tumors are said to have cancer, and the spread of cancer beyond its original site is called metastasis. Again, those are all important things for your everyday life, because sadly one day you're probably going to have to deal with cancer. But this is not a health class or an anatomy and physiology class, so I'm not going to ask you all those questions on the exam. There might be questions like that on the study guide. Here's a picture of an example, breast cancer. So here's the tumor, the original tumor, and it's near the glandular tissue. And again, this is not anatomy and physiology class, but the glandular tissue is also connected to the rest of your body through blood vessels and lymph vessels. So the tumor spreads into the glandular tissues, then it spreads to the blood vessels and the lymph vessels, and then it gets spread throughout your body. And then your breast cancer turns into any other cancers, and as you can imagine, it is deadly. I guess when we get back on Monday, we'll talk about how we treat cancer. But for now, the last word for attendance will be invade. The word I just circled right there. Invade is the last word for attendance. Brock, did you want to take that picture? Yeah. Okay. Extra credit for that. Anybody want to take, or be in the picture, I should say. Anybody want to take the picture for the extra credit? All right, Brevin will do it. And don't forget, you guys online, go ahead and send me those keywords. Meeting for office hours, I'll be online.